Good afternoon to everyone here and to people online. So it's my great pleasure to introduce Julian Neumann from Charité University Hospital in Berlin. And I wanted to say that, like, used to be the case when people ask me what's the most rewarding part of being an academic. I used to say it's the day when you send the proofs of a paper and you know that you'll never see this paper, this project ever again. That was the best part. But I think since then I changed my answer. Now my answer is the most rewarding part of being an academic is when you see your trainees succeed. And although Julian cannot exactly call him my trainee because when he spent 4 months here with me, like exactly 10 years ago, said, but his real mentor, Andrea Kuhn, she also trained here at Green Square. So as a community, we can have and take more responsibility for Julian's career successes and for this kind of line of research that he pursues, which was started here by Peter Brown and others. So I think we can all share. So it's the best part of being an academic who's been here tonight. So welcome, Julian.
Yeah, thank you very much. Thank you very much. It's an honor actually to be here. So I used to work here and come to the brain meetings and saw all these brilliant people talk. So coming back here is feel it's really great. So I'm going to talk about invasive brain-computer interface for Parkinson's disease, and just this is kind of a reminder that deep brain stimulation itself is actually now becoming a device that is capable to interact with the brain and requires computations and algorithms and is essentially now a brain-computer interface. Before I start, let's see. Before I get into the depths of this, I'm hearing impaired, so if you have questions, please speak loudly. Don't take the stuff I'm saying, not everything too seriously. All the work is done by a huge team, so this is not my own effort, as Vladimir already said. Basically, there's a whole family of people between Oxford, London, Berlin, and many other centers, and there are so many people who have contributed to this field, and it's impossible, unfortunately, in this short time to actually acknowledge all the contributions. So this is just, my kind of window into what has been going on in our surroundings. So subdynamic deep brain stimulation is a clinically effective brain stimulation technique, and that I think makes it a very unique research tool because we actually have almost 100% response rate in Parkinson's disease. So if you have a patient and implant them with deep electrodes, they will improve. So that makes it a reliable way to interact with the brain and study its functions and the role of the circuits that we are affecting. And the circuits that we are affecting are dependent on what we want to treat, but mostly the basal ganglia. So the most common target for Parkinson's disease is the subdynamic nucleus, which is shown here on the top. Can you see my cursor? No, but I can change to a laser pointer. So yeah. So the subdynamic nucleus is this structure here. You can see it on the post-mortem MRI as a hyperintensity in the T2 image, just below the internal pallidum. And the electrodes are implanted and connected to a pulse generator. And then practically the stimulation that is applied is quite simple and consists of a pulse shape that is rectangular. The shape, the actual duration of this pulse is relatively short, so it's somewhere between 30 and 120 microseconds. The amplitude depends on the effect, so you titrate the clinical effectiveness with the patient, and the frequencies are commonly above 100 hertz, most often 130 hertz, and that is kind of an empirical frequency that has evolved as an effective frequency. In some cases, or specific to symptoms that you want to treat, you may change this frequency to lower or higher alternatives. The total energy that is delivered is kind of a combination of all these parameters, and these parameters actually affect the way we interact with the neural circuit. What is really unique about deep brain stimulation is that we have a direct access to the human brain. That means we can basically zoom in and out to in between the micro and meso and macro scale, record single units, for example, in humans, and see how the circuit where the electrode is implanted responds to the stimulation. And you can see that, for example, in the thalamus that receives primarily glutamatergic afferents, a single pulse will lead to a dramatic increase in spontaneous firing just because we are exciting afferents, excitatory inputs to the structure itself. However, if we stimulate at very high frequencies, we can suppress the neural activity in these structures. So deep brain stimulation is essentially suppressing local firing as long as you have a high enough stimulation frequency and stimulate for a long enough time. So the concept is that if you stimulate long enough, you probably have synaptic depression due to vesicle depletion, and you are not activating the neurocircuit, but you're disrupting the neurocircuit. And that is even more dramatic in GABAergic targets, where even the single pulse already leads to a suppression of local firing. And for example, in the internal pallidum or in the substantia nigra pars reticulata, that receives up to 90% inhibitory input. So, but this is when you record single units or neural activity in the directly vicinity of the target where the stimulation is applied. Of course, these synaptic connections are much more complex, but can also be studied. And for example, for the subthalamic nucleus, there is a hyperdirect pathway from cortex directly to the STN that is glutamatergic. There are also GABAergic afferents, and at the same time, there are recurrent networks in between these structures, and you can basically dissociate a lot of these synaptic effects at the microscale by recording, for example, from cortical neurons, and you can see that some of these neurons can be excited, some have like a very short antidromic activation and suppression afterwards, and some neurons are totally suppressed. And the similar levels and the recurrence of these synaptic connections can be shown in the deeper structures, where an initial activation through excitation of glutamatergic neurons is followed by suppression that is basically a volley of antidromic and autodromic activation in these nuclei. Ultimately, these responses become very complex very quickly, and it can make sense to look at higher numbers of neurons and neuropopulation activity, and that would be the mesoscale. And we can record and have done that, and Vladimir has a lot of experience doing that. Record neural activity from the DBS electrode itself as local field potentials. That is very similar to an intracranial EEG from stereoencephalography in epilepsy patients. And doing so, a lot of people, including us, have shown that there is a neural synchrony in the beta frequency band in Parkinson's disease that is excessive in the circuit. And when we apply deep brain stimulation, so you can see here's the DBS is off. We are recording from the DBS electrode. There's this increased synchrony. When we turn the stimulation on, this like excessive synchrony is suppressed. And here you can see a time frequency spectrogram, frequency here on the y-axis and time on the x-axis, and below you can see the increased stimulation amplitude that we apply directly in vicinity to the recording electrode. You can see that this peak in the 20 Hertz range in the beta range gets suppressed in dose response with dose response relationship, basically more suppression, more DBS amplitude, more suppression of beta activity. At the same time, you can also look at evoked resonant activity. So every time we stimulate every pulse at 130 Hertz, so 130 times per second, the neural circuit is generating a response to the pulse. And you can see that here. So this is basically zoomed in to a series of stimulus pulses. And what is very interesting here and not explained yet is that there is even like a resonance that goes beyond the actual stimulation time. the amplitude of this resonant activity again follows a dose-response relationship. So here you can see the time after pulse in milliseconds, and these are just repetitions of the same stimulus being applied at different amplitudes, and this is showing an increase with amplitude, this response. So beta activity and this resonant activity can be related, but they don't follow the same rules. For example, beta activity is suppressed with higher intensity, and the resonant activity is increased with higher stimulation intensity. But for example, during sleep, resonant activity remains, while beta activity is suppressed, even if stimulation is very low. So what this tells us is that beta activity is an intrinsic activation pattern of the neurocircuit that is being active, while the resonant activity is a response, a passive response to the external stimulation that is applied. And we can actually follow this passive response not only in the neural field recordings, but also with single units, and see that there's actually a sequence of excitation and inhibition at the circuit level that can be followed in human patients. And this is very, very, very interesting because it allows to try and translate the synaptic level to whole brain circuits, and allows to try to understand bigger picture changes in brain network activity. For example, by looking at evoked responses also on the cortical level with MEG, looking at changes in oscillatory networks on, for example, Vladimir and others have shown that beta activity at the cortical level can be suppressed through subthalamic stimulation. And that is demonstrating the circuit effects that these local changes in synaptic activity can have. There's changes in phase amplitude coupling. So this is a report from Phil Starr's group in Nature Neuroscience 2014, I think, and where they showed that beta gamma phase amplitude coupling at the cortical level is increased in Parkinson's disease, and stimulating in the deep structure in the subthalamic nucleus actually suppresses phase amplitude coupling in cortex. But also MRI network activity can be related to some of these changes that we can study at the local level. So ultimately, the main point is that we have we can zoom in and out of the scales of brain network activity with deep brain stimulation, from single unit firing to local field potentials in deep structures to cortical subcortical coupling. And that is followed by the clinical effect of DBS that can be quite immediate. So this is a typical patient from our clinic. He shows the signs of Parkinson's disease, which are mainly slowness of movement, bradykinesia, the struggle to initiate movement, akinesia, increased number of step size, increased numbers of steps while turning, but also struggle to initiate gait and walk. You can see a typical sign, the absence of spontaneous movement of the arm while walking. This is a very typical Parkinson's disease patients. Tremor is often also associated with Parkinson's disease, but it's less disease defining. And if you turn on DBS, you can see that this patient's ability to move is dramatically improved in over a very quick period of time. So this provides us not only with the methods that can allow us to, study the measure brain activity, but also to relate clinical changes and behavioral changes to the changes in brain network activity. And that has led to DBS being a very successful research platform where many new concepts have been developed, how this can emerge and can use neuroscience methods, human neuroscience methods, to improve our ability to treat these patients. And that starts from preoperative planning. So you can use connectomics and start to actually target networks instead of local structures, and there's a lot of development in that. Then intraoperatively, we can do the single unit or multi-unit recordings, but can also start to basically merge all the information that we have from neuroimaging, neurophysiology to get like an atlas-based approach. I'm going to show an example of that very soon. And then we can use the neurophysiology to actually improve how we treat the patient, how we make clinical decisions for these patients, and ultimately design a brain-computer interface that reads brain activity and adapts the stimulation to the individual situation our patients are facing. And that is kind of the target that what I want to get to today and what the future holds for us in that regard. But first, to show you the concept of the surgery itself. So this is a very nice video from Simon Oxenford. He is a PhD student in Andy Horn's lab, also affiliated with us. And you can see that the neurosurgeons kind of implant multiple microelectrodes, and these have like 10 micrometer tips, so they can record multi-unit activity that you can see here. While the electrode reaches the target structure, the neural firing is being increased. At the same time, it's possible to co-register the native space MRI, but also different atlases. And you can kind of create a summary map of the neural firing patterns in 3D space. And this is actually an MNI space in real time, because the preoperative image has been normalized before the operation. And in the OR, you already have the normalized image available and can transform native to MNI space. Then you can decide which trajectory is most promising. And you saw that the back electrode had the most neural activity. And then you can use electrical stimulation, visualize which fiber tracts you're activating. In this case, this is the corticospinal tract, which can produce side effects. So if you stimulate too much of the corticospinal tract, then you will have direct muscle contractions. Those, again, are recordable with EMG. So you can actually intraoperatively use all the modalities that we all know from human neuroscience and really develop something that helps the patients and improve their outcomes. For neuroimaging, there are many, many techniques that people have been developing, and especially Andy Horn, who has also presented here before. And we have been trying to combine neuroimaging and neurophysiology by, for example, looking at the spatial distribution of neurophysiological patterns that we recorded, but also relating them to atlases or connectomic fiber tracts from normative connectomes, and relating the improvement, for example, or behavioral changes with stimulation to whole brain networks. What I want to talk a little bit more now is the future of the neurophysiology-based advances in DBS. And there, the main concept that is dominating right now is the idea of adaptive deep brain stimulation that uses a neurophysiological biomarker to get a readout of the current necessity to treat the patient. And that means you need some sort of neurophysiological signal. Typically, it would be the local field potential from the DBS electrode. You measure a biomarker activity, such as beta oscillations that I've introduced before. You have a signal processor that does filtering, epoching of the data, Fourier transform, smoothing, all these things. You have to define a threshold, and then a control policy will turn the stimulation on and off according to the presence or absence of beta power. And this all really builds on the work of Peter Brown, who used to work here and has shown hundreds of times, and we have reproduced even more often, that there's this excessive amount of beta activity in the absence of dopamine in Parkinson's disease. And we can disrupt and suppress this activity not only locally, but at the network level. And this is a really nice paper by Ash Oswald. You probably also know here also with Vladimir from 2016 that showed that DBS can suppress beta local beta activity, but also beta network synchronization. And this suppression correlates with the clinical improvement of the patient. So the concept is from a proof of concept that was shown here in 2013 from Simon Little, that used like an external laptop, has been translated into a new device that is called Medtronic Percept. It's the first commercially available brain-computer interface for Parkinson's disease, and that can record this brain activity and can adapt stimulation to this activity pattern in real time. So I think this is a huge translational success that comes actually from this very location. And I'm very happy that I've become part of this journey. So this is a patient in our clinic in Berlin, implanted with the Percept PC. And you don't see anything special with this patient. It's a Parkinson's disease patient. You can almost see that immediately based on the lack of spontaneous movement and slowness of movement. So what is really special is that we now know exactly where the electrodes are. So this is a patient who has electrodes in their brain. You can record the behavior, the clinical response, the neurophysiology while stimulating. So we just increase the stimulation from 0 to 1 milliamp, turn it on. Can see the beta activity respond in real time while we observe the patient. And when increasing the stimulation amplitude, you will see that the movement is getting better. So it's getting faster. The patient is more able to reach velocities or decrease the bradykinesia with higher amplitudes. And at the same time, we can follow the beta oscillations and how they are being reduced with stimulation. At some point, when the stimulation is too high, you will reach side effects such as paresthesia or muscle activation through activation of the corticospinal tract. And then you may have to reduce the stimulation amplitude again. And what you can see, this is where we are now. She tells us now that she has paresthesia in her left arm. We turn stimulation off, and you will see in this time frequency plot the beta activity coming back with almost immediately. Now here it is back, very strong beta oscillations in the subthalamic nucleus. So this is an unprecedented level of invasive neurophysiological detail that we have that we can study in humans now. And this is on one hand a huge opportunity. On the other hand, it also can overwhelm the clinicians because you already have a lot of parameters that you need to choose from. For conventional DBS, you have to decide which exact location you want to stimulate, at which frequency, which pulse width, and which amplitude. With adaptive DBS, there are additional parameters, for example, from where exactly you want to record from. So you can decide where you stimulate, but you also have to decide which recording montage you use. Then you have to observe the spontaneous activity, and you have to decide on a neural frequency that you want to use as a biomarker. And finally, the most important but also most difficult choice you have to make is which control policy you are going to use. So the one that Peter has and Simon have proposed was that if beta is high, you immediately turn stimulation on. If it's low, the stimulation goes off. But there are also alternative approaches, for example, that the stimulation is not changed as long as it's in between, the beta power is in between two thresholds. If it reaches above the threshold, stimulation goes up. If it reaches below the lower threshold, the stimulations go down. But there could also be a proportional control that is implemented in a different device called from Neuronica company in Italy. So essentially, you kind of have to develop control policies that cater, like take into account the neurodynamics and the temporal dynamics of the neural biomarkers that you use, but also the changes in the clinical state of the patient. And this is a little bit complex here, but the essence is that Parkinson's disease patients usually take medication, and then their symptoms go away, but over time they come back. So a PD patient with severe Parkinson's disease can take up to 8 or 10 doses of medication. So every 2 hours, they kind of roller coaster through presence and absence of symptoms. And then the control policy basically defines how it acts in response to these symptoms. And you need a very, very well thought through approach to define how you want to tackle these roller coaster rides of symptoms of PD patients. So this requires a thoughtful approach from like having an assessment of the clinical profile, having assessment of the neuroactivity that you can record, also the symptom biomarkers that are present in the individual patient, make sure that there aren't any artifacts that can be problematic. For example, ECG is a huge problem for implantable devices. The heartbeat is such a strong electrophysiological activity that it can often contaminate neuron recordings. And then once you are sure that you actually have a signal, you have a symptom that you want to measure, you can start tuning the control policy and adjust the parameters that you believe can help the patient individually. So this is currently under investigation in multi-center clinical trials. And it's kind of out of the hand of the individual lab, which is good because it's overwhelming. It's not like an experimental approach anymore. It's a company-sponsored, large-scale, international, multi-center trial that's going to try and answer these questions. So there's a proof of concept that we can control neural circuits at millisecond precision, which I think is a huge advance. Previously, if you take a drug, it takes a lot of time to get into your system. It passes the liver, it has to go to the brain. Everything is like really slow. But with brain stimulation, you can actually interfere with brain activity with an unprecedented spatial temporal precision. But the clinical utility has to be proven. Now, I am part of an academic experimental neuroscience lab. So I leave the beta-based adaptive DBS to the multi-center trials, and I'm thinking a little bit further about the relationship of dopamine and Parkinson's disease and how we can actually adapt the stimulation in a way that can support the neural circuits instead of disrupting them. And something that I figured is a little bit problematic in this approach of beta activity being the measure is that we come from the concept that Parkinson's disease is associated with loss of dopamine. But at the same time, we also know from non-human primate experiments, but from different sources, that there is a relative negative correlation of beta oscillations and dopamine release. So that means when we use beta activity as a trigger for DBS, we always stimulate when there's a phasic dip in dopamine. But I don't think, so my concept or my thinking is, shouldn't we support what the intrinsic system is trying to do? So when there's a phasic decrease in dopamine, probably it isn't the best time to release release dopamine or support the system. Instead, I would say that if a Parkinson's disease patient has remaining dopamine, it should, the stimulation should actually support that and stimulate when beta is low. So a neuroprosthetic control would actually do the opposite. And I'm still struggling a lot with different people from different fields about the role in dopamine. I'm discussing this, but what has really emerged over the past years is the fact that dopamine is not reward prediction errors. Dopamine is much more than reward. And there are more and more papers, also many projects here at UCL that demonstrate that dopamine is being released with movement itself without any reward being necessary. And I believe that there is a good reason for that, and that we can approach DBS potentially at some point as a support for phasic and intrinsic dopamine release. I'm going to explain that a little bit further. I just want to point you to this review from 2018 that already makes the point that dopamine is released with movement. And this preprint probably out now, showing that in the dorsolateral striatum, dopamine is not related to reward at all, but to actual movement. So if we have this loss of dopamine in Parkinson's disease, and we have all these signs, motor signs and non-motor signs in Parkinson's disease, how can we use DBS as a circuit intervention to try and support the patients and their brain activity in a way that reinstates physiological communication? For that, it's important to kind of find a good answer to the question of what do the basal ganglia do? What does Parkinson's disease, the loss of dopamine in the basal ganglia constitute in terms of the functional consequences? And there is the whole rate model of the basal ganglia essentially led to the idea of having an indirect stop pathway and a direct go pathway. And the idea that is quite convenient is that if we disrupt the subdynamic nucleus, we basically disrupt the stop pathway, leading to more ability to move. Practically, there is a lot of evidence against this simple go, no-go paradigm. And one particularly interesting study that I like very much is an optogenetic closed loop paper that implanted optogenetic fibers into the striatum in mice, which perform joystick movements. And the stimulation was triggered by movement velocity, and the direct pathway or indirect pathway were activated by either very fast or very slow movements. And what is very interesting is that if you stimulate the very fast movements of this mouse performing the joystick task, the direct and indirect pathway activation follows the concept of go and no-go. So direct pathway is the go pathway, stimulating during fast movements increases movement velocities. Indirect pathway is the stop pathway, stimulating during fast movements, the movements get slower. But if you stimulate the opposite, only the slow movements, this can invert. And suddenly the stop pathway can increase movement velocities, which is entirely incompatible with the whole concept of go, no-go and inhibition as the basal ganglia, as the function of the basal ganglia. So I'm trying to develop new ideas how this general concept of the stimulation being dependent on the ongoing behavior can be translated to Parkinson's disease. And for that, the concept of neural reinforcement can help. So this comes also from the reward domain. It's a very nice review article by Rui Costa and some of his people. And it basically states that neural reinforcement is behavior reinforcement, and it relates to the likelihood that a certain neural population dynamic emerges and re-enters the circuit. So in times of high dopamine release, the ongoing neurodynamic will be strengthened and will be more likely to re-enter and reoccur. In the absence of dopamine, the opposite can happen. The ongoing neural population dynamic is weakened and may dissolve with repetition. So this is kind of a penalization of the neural circuit of ongoing activity, and that will lead to a loss of the behavioral consequences. Now we can understand or potentially try to understand Parkinson's disease of a state of chronic suppression of reinforcement, where you have the direct pathway and the indirect pathway in a physiological balance of reinforcement, and the loss of dopamine leading to an excessive activity of the indirect pathway in Parkinson's disease leads to a state where every ongoing neurodynamic is being penalized. And if you translate to the basal ganglia circuit, you can imagine like a dynamic activity getting into the cortical basal ganglia loop and coming out weaker and weaker. And practically, this is clinicians may be reminded by that by the the most disease-defining feature of Parkinson's disease, which is called the decrement of amplitude and velocity. When Parkinson's disease are asked to tap with their fingers, what is defining Parkinson's disease, especially idiopathic Parkinson's disease associated with dopaminergic loss, is that with every repetition, the movement is getting slower and smaller. So it appears that exhibiting the movement itself has a negative consequence of the future movement, which is nicely explainable by this concept of negative reinforcement or penalization of neurodynamics. So how can we reinstate this neuroreinforcement with adaptive DBS? So the first very principal idea that we can try is to conceptualize DBS similar to the optogenetic activation. And I told you in the beginning that DBS can suppress local activity of the subthalamic nucleus. The subthalamic nucleus is the most important structure of the indirect pathway. So if we suppress the indirect pathway, we shift the balance towards the direct pathway, the putative goal pathway, and may be able to reinforce behavior in real time. So we are trying that in the proof of concept study by reproducing the optogenetic behavior in human patients, and we are measuring movement kinematics in real time and either stimulate particularly fast or particularly slow movements. And we demonstrate that in the first proof of concept that indeed the stimulation effect depends on what the patient is doing at the time the stimulation is applied. So if we stimulate during fast movements, the movements get faster, but that's not the case when we stimulate during slow movements. That means suppression of the pathway doesn't just increase movement velocity. It depends on what the patient is doing at the time the stimulation is applied. And that is entirely not considered currently for any treatment like that. And I think we should really start thinking more about how we interfere with the circuit in terms of what the circuit is actually doing during the interference. So we hope to be able to use that in the future in the concept of embedded brain-computer interfaces. And for that, it will help to use cortical activity. And Philip Starr here, the most upfront article from many recent publications, high-impact publications on brain-computer interfaces, shows the possibility to actually implant 2 ECOG strips and at the same time record local field potentials, and then you cover basically the whole circuit. So what we do through a collaboration with Mark Richardson, he's Director of Functional Neurosurgery at MGH at Harvard University, is we perform the combination of local field potential and ECOG recordings in our patients in an externalized way. And we can, for example, measure the impact of dopaminergic medication, not only on the subthalamic nucleus, but also on invasive signals of cortex, demonstrate again, reproduce that dopamine suppresses beta activity, in this case, not only in the SDN that is shown here, but also in motor cortex and generally in sensory motor cortex. We can also show that Granger causality is suppressed with dopamine, where cortex drives the SDN, and this addition of dopaminergic medication actually reduces the subthalamic vulnerability to cortical synchronization. But ultimately, what we want to get at is an approach where we can use multivariate brain signal decoding to specifically support intended behavior. And this is like super conceptual perspective thing. We're working toward that, but of course, you will have many doubts regarding like, how can we know when we need to stimulate? And I completely agree with that. I'm just saying that in advance. Generally, the concept is we have cortical ECOG activity, which is extremely high resolution, high fidelity, and we can apply machine learning methods to predict the current state the patient is in and use that for a millisecond precision intervention. And we have developed the toolbox for that in Python. If you're interested in feature extraction for machine learning applications from neurophysiology, this can be interesting for you. It's more suitable for few channels, so not ideally optimized for MEG, but practically it can be used for that. So you would have a signal stream, and what is really great about the toolbox and what we needed was that it can actually simulate the real-time situation, because a lot of decoding studies use methods that wouldn't work at all in a real-time environment. For example, normalization, Z-scoring, baseline, artifact correction, all these things are usually done before, and then you say that you can have a significant prediction. But in a real-time scenario, that wouldn't work at all. So we developed something that really simulates the real-time environment, and we can use a lot of different features, re-referencing in real time, artifact rejection in real time, oscillatory features, waveform shape features like waveform shortness, asymmetry, or other more recent developments in that field. Also a periodic component of the activity from FOOF, and then nonlinear dynamics of the signal itself, and projected to space and have some relationship between the location, the brain networks, and the therapeutic delivery. And we have a few use cases where we show the utility of the toolbox. First one is another field where invasive brain-computer interfaces are being used now, and that is for epilepsy. This is the RNS NeuroPace device that can have one sensing electrode, one stimulation electrode, and we basically benchmark our toolbox by comparing the results that you can get when you take the signals offline and try to decode the presence of seizures versus the onboard platform. And we can show that it's possible not only to suggest how you should set up your device itself, but also you can dramatically extend the numbers of features and the ability to decode seizures compared to the few features that are available on the embedded system so far. Then we also looked into emotion decoding. This is invasive neurophysiology for patients with depression that were undergoing a clinical trial with the stimulation in the anterior cingulate cortex. And they were participating in a picture viewing task, a passive emotion task. And we could use our toolbox to mainly use high frequency activity and waveform features to decode the presence of emotional versus neutral stimuli. And then we can also look at the brain networks where the decoding is most effective, and use that in the future to define targets, not only for stimulation, but also for decoding. Now, what I wanted to get at more is the movement decoding, because that's relevant for Parkinson's disease. So we trained our system to detect movement. And what is really problematic so far and not very well taken care of is the fact that every electrode location is different in every patient. So it's not like MEG or EEG where you have the standardized location, so you know which channels or how to do like a cross-patient decoding. But I think a decoder in a device that is supposed to be clinically adopted should be working out of the box. So I don't think patients should come to the clinic and do like movements and train a model before the device works. I think the device should work without any further training on the individual level. And that's why we came up with an idea of identifying the whole brain MRI network that underlies a certain location of recording and store the information of how well you can decode movement based on that network. And then you can use that in a real-time setting and check which of the available contacts match that MRI network where you can optimally decode movements from. And this allows for an automatic channel selection for any decoder, and you can train models actually across subjects. And that's what we did here with 16 PD patients from Pittsburgh, 12 patients from Berlin, 10 patients from Beijing, 18 patients from Washington with epilepsy. And what we could show here is that we can, of course, at the single subject level, decode the presence of movement. And that is in the range, I would say, of at the single sample, 100 millisecond precision of 80% correctness. If you take movements, individual movements for Berlin data, we get 100%, which gets towards useful accuracies in the clinical realm. What is important to note is that with disease progression, the decoder gets worse. Another aspect that we should be aware of, that if you have a brain-computer interface and you are recording brain activity from a brain that has a disorder, the output of the decoder may be affected similarly to the behavior, actually. So that's what we observed and reproduced actually already the second time now, that in Parkinson's disease, the disorder is affecting the brain signal decoding performance. So, but now we have a decoder, and this is our first patient recorded 2 weeks ago, where we did not train anything. That was the pre-trained model, and we just record in real time. This patient sits there, and the decoder can actually tell us in real time when the patient is moving. So this is the actual movement, and this is our decoder that hasn't seen the individual patient's brain activity yet. So I think this is a huge step forward for us towards a brain-computer interface that can support movements, stimulate in relation to movements without requiring any further training. And we may be able actually to stimulate before the movement occurs. And to study that, we have a different study that is like offline, where we look at the movement intention period. Patients are asked to self-initiate movements, and we observe brain activity before the movement starts. This is ECOG time frequency plot. This is SDN time frequency plot. These are readiness potentials similar to EEG. And we can see that in the medication off state, the readiness potential starts much earlier than in the medication on state. So it appears that there's a longer progression from initial starting point of the movement to the actual movement execution. And to study this at the single subject level and be able to compare that on a group level, we again trained brain signal decoders and on the movement intention period, and looked at how early we can actually decode movement intention. And again, we see that there's a significant effect of DBS and medication, that is here in green and red, on the time point. So the accuracy is not significantly different. All decoders had high decoding performance, but what is really significantly different is the onset of the decoder output. And that implies that there is a delay between movement intention and execution in the absence of dopamine. So dopamine and DBS can reduce the time point that the patient requires from intending to move to execution, to executing movement. At the same time, there is a change in brain circuit activity from cortical drive of beta to the SDN to cortical drive of theta to the SDN. It's very interesting because theta activity has been related with hyperkinetic movement disorders. Tremor is in the theta frequency range, but also dystonia. There are many disorders. If you look at the brain activity in this low frequency range, it is usually associated with more movement. So it appears that on a dynamic level, even in the motor intention period, levodopa as an intervention, but also subthalamic DBS can shift the circuit dynamics from a antikinetic or inhibitory state to a prokinetic movement supportive state. And I hope that in the future we may leverage that to create a neuroprosthetic that shifts brain activity to support the patient when they require it. So with that, I want to finish and thank my lab in Berlin and our collaborators, particularly Mark Richardson, neurosurgeon, who introduced me to ECOG, Andrea Kuhn, who's the director in Berlin of the Movement Disorders Neuromodulation Unit, and we do all our work in collaboration. And then Andy Horn, who is now in Boston, and the remaining clinical team and the funding agencies.